I'm McFarlin, and this is my poster, Life is Better Together. Isolation of a cold culture of bacteria from the human oral microbiome shows a combination not ever seen before. So I'm very interested in medicine, so I wanted to focus on the human body. So I specifically chose the oral microbiome, where it's known to have between 500 and 700 different microbes. Common players are Streptococcus and Staphylococcus. Primary functions are to aid in protection of your gums and your teeth, and they also form biofilms, which just produce a layer in your oral microbe to help trap different particles. And so, like I said, I'm interested in medicine, and I wanted to get an antibiotic-resistant microbe. So what I did, I just took a sterile cotton swab, and I swabbed the inside of my gum line, and then I transferred it to a Mueller-Hinton agar, which is known to help grow antibiotic-resistant strains. So after doing that, it didn't grow very quickly, so we ended up transferring it to a TSA plate, which grew exponentially after that. So here you can see a picture of just one of my subcultures. You can see the colonies are small, the cells are relatively the same size, and it's a deep yellow. So from that, I made a wet mount slide here, stained in crystal violet, where you see the cells are less than 1 micrometer in diameter, where the colonies are smooth and circular in form with an entire margin and flat elevation. And so from that, I then furthered into an antibiotic susceptibility assay, where all these white discs are antibiotic impregnated discs, where I tested for 5 common antibiotics down here. And so as you can see, this white one is a control, and it's the only area where my bacteria grew, which shows that all the other antibiotics my microbe was susceptible to.
That's a pretty weak bacteria.
Oh yeah, very weak. And so then we isolated the DNA and we sent it over to UNH. And then once it was all inputted into ROD, we ran through Termomatic and Spades, and then using Proco we looked for a 16S gene. And from that I got 2 hits, so it means I had a cold culture with 2 different microbes. And I had a Streptococcus salivarius, which is the family tree here, it's highlighted in yellow, and then a Dermacoccus nichomyenisis, which again here is down in this family tree, with again highlighted in yellow. So from that I wanted to just get a little bit more confident and be sure that those are the 2 that I got. So I ran it for common housekeeping genes. I ran for D-alanine ligase, superoxide dismutase, also known as SOD-A, and DNA gyrase subunit A and B, where for all of those I got both the Streptococcus salivarius and Dermacoccus nichomyenisis. And so with that, I then started to do my research, and I found out that Streptococcus salivarius is one of the first microbes to form after birth in your mouth. And it's thought to play a role in your immune health and help in the inflammatory response. It also synthesizes extracellular glucotransferases, which again just help different particles and stuff in your mouth. And the gram stain's positive, which you can see here. And then with the Dermacoccus nichomyenisis, which very interestingly, it was from what we researched, it's the first report of finding it in the human mouth, which freaks me out a little bit, but it has been seen on human skin, tap water, and deep ocean sediment. So we're thinking it might have come from the tap water I drank. We're hoping it did. And it also gram stains inconsistently. So again, back to this picture, you can see little pockets of purple with the gram negative. Originally, we just thought they were little pockets of the stain and the dye, but it turned out I had a cold culture. So to conclude this, again, this is the first report of the Dermacoccus nichomyenisis in the human mouth, and it's also the first report of it in a cold culture with a Streptococcus salivarius. And then in the future, we want to separate the 2 genomes and do further analysis, the genomic analysis and the phenotypic analysis, to see if maybe they have a mutualistic relationship.